Welcome, my name is Arna Christensen. I'm a faculty member in the biology department at Westfield State University. And this is the first of three videos that are intended to help you to prepare for the biology MTEL. The first video will look at some of the general kind of structure of the exam, as well as some of the subject matter, and then lead into some sample questions. The second video will look at more of the subject matter and sample questions. And then the third video will provide some strategies for some of the different types of questions you can expect to see, as well as sample questions. So today we'll be looking at some of the general information, some of the subject matter starting with the chemistry of life and molecules, and then looking at the sample questions. You'll have been provided access to an online folder that'll have some resources available to you, and of those resources, these PowerPoints that I'll be going through, or these slideshows that I'll be going through, you can download them because they have some live links that'll take you directly to the different types of resources. But some of the examples that you have are Westfield State University's resource guide for the MTELs in general. There are some links here that take you to the Massachusetts MTEL website, which has sample tests as well as an enormous amount of information. So one of the things you'll definitely want to do is spend some time going through the Mass MTEL website, specifically for the bio subject matter exam, and see what they have to offer for some of the details that I won't be going over. Praxis is a company that you can use to access some sample exams if you want. And then also here's a book that I recommend that has an excellent overview of a lot of the material, the subject matter, to help you prepare. So it's named MTEL Bio 13, Teacher Certification Test Prep Study Guide. So the Bio 13 is the previous number that was used. So now you'll see it referred to as 66. But the vast majority of the content overlaps, so I still recommend this book. To register for the test, you'll have to go to the DESI site, right? So there's a link to the site there. You'll need to first get an ELAR profile, as well as a Massachusetts Education Personnel ID. Give yourself some time to prepare for the exam. Like with anything, the more time you have, the more opportunities you give yourself to go over the material, the better you'll be able to retain it and the less stressed you'll be for the exam. The actual exam is 100 multiple choice questions and 2 open response questions of 4 hours and 15 minutes. And the links are here to sign up. So just so you kind of know what to expect, if you're thinking about a particular month that you want to take the exam, by going to these links, you can expect to see, 1 or 2 days within a month at these various different centers where there'll be slots available for you, if you give yourself enough time, of course, to book ahead of time. For multiple choice, there's 4 responses to each of the multiple choice questions, A through D. They count equally, each of the multiple choice questions, and there's no penalty for answering incorrectly. Right? So this is certainly one of those cases where you want to answer the question, no matter what. The open response questions will give you an opportunity to provide either handwritten or typed responses, or a mixture of the two. Right? So I would probably plan, because my handwriting is not very good, I would expect to type my responses, but then use the opportunity to draw some illustrations or diagrams or hypothetical data that might be relevant to the open response question, and then scan that in and include it as part of my response. The DESI MTEL site has an awesome set of examples of open response questions, as well as how they were assessed given a strong or weak response. So that was an excellent use of time to go through and get a sense of not only what the questions will look like, but how they're going to be assessed so that you can be strategic in your response. You want to recognize that the assessor, and I'll spend some more time with this on the third video, is going to be looking at whether you've achieved the purpose, right? So how well have you responded to the direct intent of the open response question? How accurate is your information in your response, right? So that's the subject matter knowledge. And then a compelling response will have additional supporting information, right? Or perhaps examples, right? So this is what will be looked at in terms of support as your response is being assessed. And then the rationale is to look to see how well it all kind of comes together. And if the open response is looking, for example, ways that you might approach instruction for a particular type of phenomenon, how appropriate is your selection of your exercise or your activity in addressing the intent of the open response. So how well does it kind of come together, and is there some rationale behind the flow of your response? In terms of preparing for the exam, right, make a plan for yourself. I know if I'm studying for something, I need to give myself time, but then also expect to be able to use different types of environments to study. So it works for me to move around, perhaps studying for a while in one spot, then going to another quiet space like a library or a coffee shop that I like to go to. Right, so it helps me at least to move around different spots over the course of hours, days, weeks, to really get my head around material and to make kind of the whole experience of studying as effective as possible. I like to talk to people about the material. I like to form, groups of people that can discuss the material, because that helps everyone involved reinforce what they know, but also identify and understand what they don't know. The content areas are much more detailed than what I'll go over here. So again, like many times throughout these presentations, there's links, and here the details link would take you to the DESI site with a lot more information on the specific details. But in general, we're going to be looking at the chemistry of life, metabolism, cellular metabolism, cell structure and function, putting those together into tissues, right, how the tissues work in humans for anatomy and physiology, building up beyond organismal biology into life coming together, into ecosystems and the flow of energy through ecosystems, and the effects that humans have on ecosystems. And also looking at genetics, heredity, and evolution. And then lastly, putting a lot of these different subject areas together for the open response questions, which it appears to generally be structured such that one open response question will cover molecules and genetics and heredity and evolution, whereas the other one will tend to be related to organisms or ecosystems. So very crudely, kind of microscopic versus macroscopic biology. We'll begin going through some of the actual material now, and one of the things that I want to highlight is that we wouldn't have time to go over all of the material that you'll be responsible for or might expect to see on the biology MTELs. I had developed these to be able to kind of spot, check, focus on particular areas that I expect that you might encounter, and it would be good to kind of refresh yourself on if you don't remember the details of some of these subject areas. But more than that, as we kind of survey all of these different areas of biology, recognize that you're going to feel stronger in some areas than others, and it's those areas where you feel like you need more reinforcement that you should go on to some of these other references that I've provided to you, some of these resources, to kind of fill that area out. Like my background is biochemistry, molecular, cellular biology. So inevitably, I feel more comfortable with those types of questions. And if I were in your position, I would need to spend more time in other areas of biology, like the macroscopic, ecology, organismal biology, and so forth. So just wanted to make that clear before jumping into it. And one area I wanted to mention before jumping into the kind of formal biological material is a reminder that in terms of the design of experiments and study design and components of a good and well-designed study, you have independent variables and dependent variables. So these are the types of bits of content and possible questions you might encounter that you don't want to have to struggle to remember or puzzle through during the exam with the added pressure of time. So a reminder, right? So with a well-designed study, you generally have something that changes, and you've orchestrated that within the experiment. That's part of the plan. And then you're measuring some sort of an effect or an outcome of that variation. So as part of the experiment, the thing that changes by design, that's the independent variable, right? And generally there's one or very few independent variables, and everything else is controlled for. The dependent variable is what you intend to better understand, right? So that's, how does something change within the experiment? Well, it depends, right? And that's how I help remind myself that the dependent variable is the thing that we're curious about and the thing that we're measuring, because how it's affected, well, it depends, right? It depends on the things that you're changing. So maybe as an example, for my own background, there was a period of time that I was interested in migratory fish like Atlantic salmon, that during the year, right, if their age and context is right, they'll migrate from freshwater streams, rivers, and then out to the ocean, right? And that's dependent on things associated with the time of year and season, like water temperature and day length and things like that. So if we were going to develop an experiment to better understand how the animal's physiology perhaps were changing as it was migrating out towards the ocean, right, as part of the experiment, the time of year or time, right, would be the independent variable. That's the thing that's part of the experiment. And then how the animal's physiology is changing, that type of thing, depending on what we're measuring, right, that would be the dependent variable. Specifically, we would be looking at things like a class of cells in their gills that help to balance salts. So they're better able to do that when they move from freshwater to seawater. So we would actually be investigating the abundance of these different types of cells, right? So over the course of the year, right, a reminder with the x and the y-axis. On the x-axis, that's where you would put the independent variable. So maybe that would be like the months, right? January, February, March, whatever. And then it's on the y-axis where we would have the dependent variable. What's changing? How it's changing? It depends, right? That's what we're curious about. So for our example, it might be like the abundance of this cell type that helps the animals balance salts, or whatever, right? So if you were to plot those data in the opposite orientation with the graph axes, it wouldn't look right. It would feel very unnatural. And these are things that we already kind of know intuitively, but you don't want to have to struggle with those concepts during the exam, right? So just remember, independent variable is part of the experimental design, right? It's the thing that you're planning on changing. The dependent variable is the effect, what you're measuring. And when you're plotting that, it's on the x-axis goes the independent variables, right? And then on the y-axis is where you're having your measurements of the things that you're changing, right? The dependent variable. So now with the formal content, right, we're going to start small, right, and that's with the chemistry of life. And again, this is not going to be an overview of chemistry. It's not even going to be an overview of all the chemistry you need to know for biology or for the bio MTELs. it's kind of a spot check of some of the really critical concepts. So we always have to talk about chemistry when we're talking about biology, because biology, basically at one level or another is chemistry. And we don't go through all of chemistry because a lot of the chemistry doesn't bear any relevance or importance for biology. But some of it is so critical, it's integral to everything that's happening in biology, that you really should have a strong foundation or understanding and appreciation for some of these basic foundational chemical concepts, that it's worth taking the time to go through them here. And if they look familiar and really elementary, that's great. Then you can think more about other stuff. But if it's not something that you spend a lot of time thinking about, or it's been a while, right, it's definitely worth spending some time getting your head around it again. Because without question, these are the types of concepts that are going to be on the bio MTEL. There'll be questions about, for instance, these different types of bonds. So atoms, right? There's various different types of elements, and ones that are really familiar, like carbon and oxygen and hydrogen. These are the different elements. These come as the smallest building blocks are atoms. But the chemistry of life requires putting these atoms together into really oftentimes large, sophisticated biological machines like proteins that are doing unbelievably exquisite types of things at the cellular level. We need to put atoms together to build really complicated molecules that have the capacity to do almost unimaginable types of things, right? So we got to bring these atoms together. We got to start to build these into larger structures. So this entails bonds, right? And these different types of bonds are going to form between atoms based on the behavior of the electrons, right? So atoms have a nucleus with protons and neutrons at the core, at the heart, and then they have these electrons buzzing around. And it's the electrons that are going to drive whether these atoms, interact or not. One thing to keep in mind is that what's driving this propensity for atoms to come together and interact is something called the octet rule. Octet for 8. And it's that atoms prefer to have their outer valence shell, at least number 2 and 3, have 8 electrons within that shell. That's where they're most stable. That's where they're most satisfied. So if, for example, you had something like sodium that actually only has 1 electron in its outer valence shell, that's a terrible situation, right? That is not energetically favorable. I think about it like a fly buzzing around, right? It's nowhere near satisfied. And at the same time, you have something like chlorine. And if you were to go through and look at the number of protons and how many electrons that results in, that's more depth of chemistry than we need to get into. It ends up having 7 electrons. And as I mentioned, 8 is the key number. So it's 1 short. It's not satisfied either. But this is a partnership made in heaven, right? Because you have sodium with 1 electron it's trying to get rid of, chlorine with 7 electrons it's looking for 1. One thing they can do is they can come in, right? If they can actually transfer electrons, that 1 from sodium over to chlorine, and now sodium's gotten rid of that headache, that 1 electron. Chlorine's been dying for 1. It's like, yeah, come on over, I'll take you. Right now they're each satisfied because chlorine has 8 electrons now in its outer valence shell. It's good. Sodium has gotten rid of that 1. And as it turns out, it means underneath that shell, it has 8 that have been waiting kind of under the surface. And its outer electron shell now has 8. This results in an ionic bond because electrons are charged. They have a negative charge. So when chlorine takes on that negative electron, it takes on a negative charge. And sodium has given up that negative electron, leaving it positive, right? So now you have a positive atom and a negative atom, right? These are called ions. Right? It's now a sodium ion, right? With a +1 charge, and it's a chloride ion, right? With a -1 charge. Opposites attract in chemistry. These two come together because they're opposite charges, right? And that's the nature of an ionic bond. There's another way that atoms can solve this problem, right? Or this kind of idealistic journey of trying to get 8 electrons in the outer shell. And that's by sharing electrons, right? So here we're not talking about a transfer, we're talking about a sharing of electrons, where you could have these two chlorine atoms coming together. Each of them are short, right, to meet that octet rule, but they can decide to share some of what they got, right? So that at any one moment in time, right, with those electrons that are being shared, they have 8 within their outer electron shell. So they're still satisfying that rule. They're still getting those 8 electrons, but to do that, they need to share. This is really critical because sharing of electrons and what's now we call a covalent bond, these bonds are extremely strong. Ionic bonds are, relatively speaking, kind of weak. And it's important to think in biology that strength doesn't mean valuable or better, right? In biology, you need strength, but you also need fluidity and integrity, like the ability to kind of quickly grab and release and move things around and have fluidity, right? So you need both kind of types of bonds to make life work. So sometimes you're really dependent on, let's say, DNA, right? DNA is a long molecule that you find in the nucleus. It's the molecule of heredity. You need integrity there. That has to last. That's the code of life in the heart of the cell. So you have covalent bonds along the backbone of that molecule that imparts strength, right? So sometimes you need covalent bonds. Yeah, but sometimes you have, interactions between molecules that are relatively fleeting and are driven by charges and not the sharing of electrons, but more about this opposites attract type of concept. And you want that fluidity, right? So that's where you see kind of non-covalent bonds coming into play. And then lastly, we have hydrogen bonds, and hydrogen bonds are the weakest of the ones we're looking at, but super important, right? So again, weakness doesn't mean less important. They're critical for life, particularly when we're thinking about the way water molecules interact. And at the core of this is if you're looking at a molecule of water, right? We have 3 of them here. It's an oxygen and 2 hydrogen. Within this molecule, there's a sharing of electrons between the oxygen and the hydrogen and the oxygen and the hydrogen. So the sharing of electrons, again, covalent bond, very strong. So within that molecule, you have these 2 covalent bonds holding together the oxygen and the 2 hydrogens. As it turns out, oxygen is really greedy for electrons. So yes, they're sharing electrons. That's true. They have to be sharing the electrons because it's a covalent bond, but it's not equal. Oxygen is more greedy for the electrons. Electrons are negatively charged. So within that molecule, the oxygen is actually more negatively charged because the negative electrons are hanging out there more. So now this molecule is polar. It has differences across it. It has more negative regions where the oxygen is and more positive regions, right, where the hydrogens are. And again, opposites attract, right? So even within water molecules, you're going to have the more negative oxygens aligning with the hydrogens because they're more positive, right? So you get these opposites attracting across molecules, right? Intramolecular interactions. And this is super critical for the biology of life, for the chemistry of life, because water is the solvent of life, right? Whether you're talking about blood or fluids in the cells or fluids outside of the cells, or whether you're talking about lakes in an ecosystem that freeze, right, and the water goes to the surface because ice water is less dense, the solid is less dense than the fluid, right? That allows organisms to thrive during the winter because the ice floats, right? All of these at every level of life, right? Cellular to ecosystem, right? This idea of water molecules and these hydrogen bonds and the way they drive the interesting properties of water are really key. You also see hydrogen bonds elsewhere in biology. So it's not just water molecules that contain oxygen that are electron greedy. Some proteins, right? Some different types of atoms, like nitrogen, as it turns out, is also electron greedy. Those are present in other molecules too, like proteins and DNA. So you see hydrogen bonds with water, but you also see hydrogen bonds, for instance, going back to DNA. We talked about covalent bonds being key for strength and integrity in the backbone of DNA. It's also the way that two strands of DNA associate is with hydrogen bonds. It's the same type of bonds that you see with water-water interactions, but it's really fluid, right? And that's key, right? You need DNA to be strong, and you get that with the backbones, but you need it to breathe and have the strands break apart, like when you duplicate DNA, right? Almost like a zipper, right? So that's a beautiful case of where you want strength and integrity with life, but also fluidity and plasticity, right? So there you got the covalent bonds, there you got the hydrogen bonds, right? It all kind of comes together. So water, you'll definitely see questions on water, right? These are beautiful MTEL questions, and you might see a couple that are related to these unique properties of water. On the side of the slide here, this is showing again, right? You've got four molecules of water, right? So each molecule of water has an oxygen covalently bound to the hydrogen, so they're sharing electrons, right? Although oxygen is more electron greedy, right? So it has more of the negative electrons around it. So it has more of a negative charge. And you can see how these hydrogen bonds are aligning with opposites attract with the oxygen to the hydrogen. The way that the molecules can arrange with this kind of structure means that in a gas, the oxygen molecules and the water molecules in kind of the vapor phase are moving around very quickly and very fluidly. If the temperature cools down, right, and you increase the density of this solution to a liquid phase, right, the molecules are going to be more packed against each other. And if you cool it down even more to turn the phase into the solid phase, the water molecules snap, aligned like we see here with the picture, into kind of a lattice that positions them farther apart than they exist in the fluid phase. So this is kind of a quirky property that you don't see with a lot of other types of molecules. Whereas you go from a gas to a liquid to a solid, like a solid you think of as being really dense, right? It's actually a little less dense based on the way that these molecules align with each other. So that's why the less dense ice, right, floats to the surface. It's these hydrogen bonds that also give water a high heat capacity, meaning that it's resistant to changes in temperature, right? So that's great if you're an organism, right? I wish that maybe it was more resistant to temperature on a day like today where it's incredibly hot, because then it means that I would be impacted by that heat less quickly, right? But whatever the temperature is outside, this property of water means it's resistant to changes in temperature, means it's kind of a buffering within life systems, which makes it a great, um, solvent of life. It's resistant to fluctuations in temperature, right? It also has a high heat of vaporization, which in part gives it the great property of cooling with evaporation, like we see as water molecules or water beating up on the surface of skin. As it evaporates, it has a cooling effect. So that can help to bring down the temperature of the surface of the body, right? Where the temperature of the blood just underneath. And as that blood moves into our core, right, bringing down the temperature of the organism. It's cohesive, so it's kind of sticky. So that gives us kind of capillary action. If you put two pieces of glass next to each other and then put that into water, you can see the water kind of move up in between. And they're kind of stuck together and following each other. That's part of this cohesiveness, right? Or a tree, right, as it loses water, right, from the leaves, right, it's drawing water up through the roots in the soil. And like a bucket brigade almost, like following each other all the way up the length of the plant, maybe it's a tree or whatever, right, as part of this cohesiveness. And importantly, it's a really great solvent because it's a little negative, because it's a little positive, and because opposites attract. It can be attracted to things that are both positive and negative because it's a little bit negative and a little bit positive, right? So because it has that more negative oxygen component, that can interact with stuff that's positive. And because it has the more negative hydrogen kind of component, it can interact with things that are kind of positive, right? So it can interact with a lot of charged things. And that means it's a great solvent. So if you were to take something like table salt from your table, right, you know it's a crystal. Um, that's like we saw before, that's sodium and chloride ions, right, that are working to satisfy the octet rule by the movement of an electron. It's these oppositely charged ions that are stuck together, and they're doing that like in a crystalline type of structure like table salt. You put that in water, right? And because water is a great solvent, the negative oxygen can interact with a positive sodium. The negative hydrogen part can interact, right, with the positive sodium ions, right? They can actually come in and start to disrupt the ions within that structure. Right, so that's shown here, right? So we have our polar water molecules, a little bit negative, a little bit positive, right? It can come in and interact with sodium ions, right, which have lost an electron, so they're positive. Chloride ions, which have picked up that electron, so they're negative. And because water is both, it's positive and negative, it can interact with both of them, and it works as a great solvent. All right, so we've talked about atoms. Atoms interact with each other based on electrons, right? These electrons can drive atoms to interact as ionic kind of components with an ionic bond, or they can work to build organic molecules, and these atoms are connected with strong covalent bonds, and we can start to build a lot of these. Organic macromolecules or large molecules, we call them organic because they're present in organisms that are alive. And one of the key defining types of elements that you find in these molecules that makes them organic is carbon. And that's because carbon, right, it needs to satisfy this octet rules like a lot of atoms do, right? There's some exceptions. But it actually only has 4 electrons in its outer shell, so it's looking for 4. So it's happy to interact in a lot of different ways with a lot of different atoms to get up to 8 in its outer shell, which is to say that carbon, right, which is integral to organic molecules, can interact with a lot of different atoms, thereby forming large molecules, right, that can make up long chains or branching types of structures, right? So we see carbon in all organic molecules. And the general classifications here are proteins, lipids, carbohydrates, and nucleic acid. Proteins, so these are the kind of tools that get the job done in cells. They can be structural, providing architecture. They can move stuff around, so they can be kind of like little motors. They can work to build up or break down other types of molecules in the cells. So they're kind of the little microscopic tools that are doing the work within cells. These are the protein. They're made up of building blocks, and these building blocks are amino acids. And there's 20 of them. And one of the amazing, incredible kind of unifying characteristics of life is that whether you're an oak tree or a human or a llama or an E. coli bacterium, right, you're working with these same 20 amino acids to build your proteins. When I pause and think about that, I just think it's extraordinary. But no matter what type of life we're talking about, I gotta have proteins to get work done at the cellular level. And these proteins are all built out of the same 20 amino acid. Right, so these amino acid building blocks together give us protein.
Fatty acids, or kind of fats or lipids, these are all kind of related terms. These are different types of structures within cells that tend to hate water, so we call them hydrophobic. So whereas proteins tend to be kind of variable and charged, and their charges can interact with the charges that we talked about in water, kind of positive, kind of negative. But you don't see those in fatty acids. You don't see those in lipids. So lipids tend to, in large part, resist interactions with water, which means that they can make great kind of boundaries within cells. So they can be components of membranes, separating different types of fluid compartments, and so forth. Carbohydrates, I think sugars when I think of carbohydrates. You can think about those as being building blocks, singular entities, which we call monosaccharides. These sugars, these monosaccharides, can be built into, you know, 2-unit structures called disaccharides, or many unit structures called polysaccharides, right? Give rise to carbohydrates. And carbohydrates are a great energy source, but they're also important kind of structural material in plant cells for sure, but a great energy source in plants and animals and other types of organisms. And then there's nucleic acids, right? So nucleic acids, they build the heritable material of life, right? DNA, but also DNA is a code. Um, Maybe you want to think about DNA as being like a library. Within that library, right, a particular cell doesn't need all of that information all the time. It just needs to check out the books that it needs to do the jobs that it needs to do. So, a cell in an eye that's receiving light signal is going to need to do different things than a cell in the gut that's absorbing material is going to have to do different things than a cell in the muscle that's contracting, right? But all these same cells in me or in a particular organism have all the same DNA. But they're checking out different books based on their needs. And that means that the DNA code gets transcribed to RNA, right, or messenger RNA. But these are built out of the same types of building blocks as DNA. Okay. So proteins, we can think about proteins, right? These are the little biological tools within cells, within organisms. They're encoded by DNA. They perform functions in the cells, and we can think about them at different levels of structure. The first type of hierarchy of complexity is our primary structure. And that's simply what amino acids, the building blocks of proteins, what amino acids are in a particular protein. Right? And that depends on the protein, right? So two examples of proteins might be like actin and myosin. These are proteins that work with each other to allow muscles to contract. They're two different proteins, so they have two different amino acid sequences. That's an important way to think about a protein and to talk about it. What is it built out of? But no protein exists like this in the cell when it's actually working, right? Because this primary structure, this simple representation of the amino acids linearly, immediately as these proteins are synthesized, and then for the rest of their existence, right, they start to take on complicated structures. And some of these structures we consider as their secondary structures, right? How are these different amino acid residues? How are they interacting with their neighbors to give rise to like a helix structure or maybe a sheet structure? And then in the reality of the way proteins actually interact, right, they come together as a higher order architecture called the tertiary structure, which is more what they actually look like in the cell when they're doing their job. Right. So as a protein is being made, it's like folding and misfolding and testing out different types of structures, and it's looking for what's energetically most favorable before it settles into its actual type of shape and structure. Right? And that's the tertiary structure. That's the way this gene product might appear within a cell. But also critically, almost never do proteins interact in isolation by themselves. right? Proteins tend to come together with other proteins, other partners, sometimes more of themselves, to be able to accomplish complicated tasks. So when we're talking about multiple chains coming together, multiple protein kind of subunits coming together, this is what we call the quaternary structure. And I'm looking at this and I'm thinking that's probably hemoglobin. So hemoglobin is a textbook example of a protein that performs a function in the body, right? So in humans, it's packed in red blood cells, right? Our red blood cells are red because they contain the protein hemoglobin that's red. Hemoglobin contains iron, which binds to oxygen, and that's how hemoglobin and red blood cells carry oxygen, right? It actually exists as four subunits coming together, four polypeptide chains. So that gives us the quaternary structure, right? So that's kind of an increase in complexity, the way we think about, talk about, and look at proteins. Some proteins are enzymes. So an enzyme is shown here in purple. An enzyme is a type of protein that generally performs some type of job. It doesn't get depleted when it does this job. It's ready to do the job again. But it might take some sort of a material, right? And we call that the substrate. And maybe it breaks it into two molecules, takes one and breaks it into two. Or maybe another enzyme takes two substrates and builds them into one. Or maybe another type of enzyme takes a substrate and just rearranges some of the bonds. Right, the enzymes are doing this type of work, and they're basically building, breaking down, and rearranging some of the molecules in cells so that the cells can get what they need access to, so they can build the things they need to build. Where substrates bind is within the active site of an enzyme. And a specific enzyme has a really specific substrate and does a really specific job. And it's amazing at this job, but it's really limited in that that's basically the only job it can do. And they're also really sensitive and finicky. So if you get in there and you change the temperature, or you change the saltiness, or you change some sort of a factor, right, enzymes are likely to become disrupted very quickly. So enzymes are amazing, great at what they do, but also very sensitive. When we talk about enzymes, we can think about, all right, so how active could this pool of enzyme actually be? If we were to give this enzyme as much substrate as we could, how much work could we really get out of this enzyme? That's what we call the Vmax, kind of the maximum velocity. And half of that is what we call the Km, right? And that's half the maximal enzyme activity. And that'll become important coming up in a moment. Right. So enzymes can be blocked in what they do, right? So there's inhibitors that can come in and actually sit in that active site and block the ability of the substrate to get in there, right? And if it does, then the enzyme, it can't do the job it needs to do. Or some inhibitors go in and they bind somewhere else in the enzyme, but usually that still affects the shape of the enzyme, so it's just not as good at its job anymore. So who cares? Well, some examples might be like drugs, right? So a lot of pharmaceuticals will come in and be able to modify the activity of an enzyme in order to be able to affect some sort of a disease state, right? So this is an important way to influence enzymes, particularly if you're thinking about biomedicine, right? And the origins and how to correct diseases. So kind of putting it all together, we can plot the activity of an enzyme, right? And I'll walk you through this, right? So horizontally, we have the x-axis here. That's how much substrate you have, right? How much of that material the enzyme is going to go to work on. And then on the y-axis here, we have, all right, so how much work is the enzyme actually doing? And if you just focus on the black line, I hope it makes sense that if there's very little substrate, if there's very little material in there for the enzyme to work on, the enzyme's basically got nothing to do, right? So the velocity is going to be really low. But the enzyme's more than happy to do its job as long as you give it substrate. So if you provide more and more material for that enzyme to work on, that pool of enzymes is going to start going to town, right? And it's going to be doing its job. And ultimately, you're going to give it so much of that material that the enzyme can go to work on all of this stuff. It's doing great, right? Most of that enzyme is going to be occupied with a job to do, right? It's going to be reaching its maximal capacity. And then, like a lot of us, right, just in life, sometimes we have too much to do and we can't get to it all, right? That's kind of the same way with enzymes. It doesn't matter how much you keep throwing at the enzyme, right? This kind of flatlines because at some point the enzyme is like, hey, I got my hands full. I'm doing great. I'm doing my job, but I can't handle anymore. And it doesn't matter how much of this substrate you throw at me, right? We're not getting any more out of this reaction, right? So that's why that kind of flatlines. So that's a normal context, right? We have the amount of substrate you can add, right? Before you're reaching this maximal capacity, and then you add any more of the substrate, you're not going to get any more out of that pool. Now, importantly, if you throw in some competitive inhibitor, and remember that competitive inhibitor sits in that active site, you're going to get less product from that enzyme because it can't do the job it needs to do. You're blocking it. So it probably makes sense that that velocity is lower. But at some point, you can add so much substrate, right? And this is where that red line now is starting to increase. You can add so much substrate that it actually gets in there and gives the inhibitor the boot, right? It gets in there and it displaces that inhibitor. There's so much of it. So now the enzyme can get back on track, right? It has enough substrate that it can get back up to this maximal kind of amount of work it can do. And that's to contrast with the non-competitive inhibitor. And because you're going out there and you're basically knocking out the pool of effective enzyme, and it has nothing to do with the active site being occupied, right? It doesn't matter how much substrate you add, the enzyme is just not as effective. Right. So this is something that I would spend some time. If it made sense the way I talked through it, that's great. If it didn't, that's also fine. I would go back and think about this a little bit more because I wouldn't be surprised. I don't think this is a for sure something you'd see on the Untitled, but this might be something you encounter. So kind of think your way through it. Use a resource if you need to, or you can always email me and we can touch base and connect and I can talk you through it again. But this might be something that you see. All right, and then lipids, right, are these things that are kind of hydrophobic. So hydrophobic molecules, right? If you look at these hydrocarbon chains, there's not a lot of variability in charge. So there's not a lot to interact with charged water molecules. So they tend to be hydrophobic. Down here, we have what's called an unsaturated fatty acid chain or hydrocarbon tail. And you'll notice with these hydrocarbon tails, they're basically saturated with hydrogen atoms. Down here, they're not saturated with hydrogen atoms. There's a few of them missing. But the carbon still wants to satisfy this octet rule, so it doubles up with a neighboring carbon. So now they're sharing electrons, and now it's a double covalent bond. And that introduces a kink in the lipids. So a kink in the lipids means that they're now unsaturated, and this can be introduced or not taken away within biological substances, and it can influence lipids and fats within organisms to make them more or less fluid, because the more of these kind of kinks you have, the more fluid the material is going to be. And phospholipids are an interesting type of lipid because they are also hydrophobic and have these hydrocarbon tails and they hate water, right? They're hydrophobic. So they tend to line up away from water, which you would find inside of a cell or which you would find outside of a cell because that's the solvent of life. They're kind of trying to hide from it. So it creates a barrier. And what's more than happy to interact with the water outside of the cell or inside of the cell are these hydrophilic water-loving heads that actually have a phosphate group that's charged. So it can actually interact with the water. And that allows these incredible substances to organize and create membranes or boundaries, as the plasma membrane of a cell, or some organelles have plasma membranes within the cell. Different types of ways to get materials across lipids can be categorized by whether they take energy or not. Passive transport doesn't take any energy. And if you have a lot of material on one side of a membrane and it's allowed to get through the membrane, it'll naturally flow from an area of high concentration to low, and we call that diffusion. Sometimes something would like to move from an area of high concentration to low, but it can't get across that greasy barrier of the membrane. It's hydrophobic, and maybe this thing is too large, or maybe it has charges. It just can't get across the barrier. But some things like proteins can give it an avenue, and they can say, yep, you are welcome to come across. We've got some matching chemistry. I'm going to give you the opportunity. It can also diffuse, but it's facilitated because a protein allows it to do it. Some things can get from one side of a membrane to another, but it takes energy because they're going from a low concentration to a high. They're going from a low concentration to somewhere where there's already a lot of it. And that is in many, many cases a requirement for life and cell biology, getting something that's sparse into an area where it's highly concentrated, but that is not energetically favorable. So it takes ATP, and we call this active transport. All right, and then osmosis is something you will probably encounter. Osmosis is an idea that water will move across membranes until there's an equilibration in concentration. And the term I use, or I have in my head to help remember this, is that water follows the salts. I also want to mention that it's 7:05. We're almost through with the content, but if you've got to duck out, that's fine. I can provide you with a link to the movie. If you want to stick with me, probably about another 5 minutes of the content. And then I can leave the multiple choice questions for you with answers for you to just work through before we meet again and go through them. So water will move across membranes because it can. It can kind of slink across cellular membranes, but it can also be given facilitated types of approaches to do that. But the idea of osmosis is that water will move across membranes until the solute concentration is equal on both sides of the membrane. And the trick I use in my head is that I say that water follows the salts. It helps me remember that water moves in a direction that's saltier until there's an equilibration. It's just important to note and realize that the reality is water isn't just following salts, it's following any solute, and that might be proteins or other types of things. But a favorite example in a textbook and elsewhere is red blood cells. And if you have a red blood cell in a solution that's isotonic, iso means the same tonicity. The saltiness is the same inside and outside of the cell. The water isn't going anywhere, right? So it's not moving. But if you have a low tonicity, low concentration, hypotonic solution, that means it's more dilute. What's water going to do? It's going to follow the salts, right? In this case, they're inside of the cell is where it's more concentrated. That's where water is going to go. And it'll continue to go in and go in and go in. And if the contrast is dramatically enough, the cell will pop. That's never like a real situation, but it's a way to kind of conceptually kind of think about where the water is going. And a student once gave me a great trick for remembering this, and it's pop. Hypotonic, the cell pops. So hypertonic would be like super salty. What if you took this cell and threw it in seawater or something like that? Where's the water going to go? It's going to follow the salts. In this case, it leaves the cell, right? And it's going to shrink and become this pathetic little useless cell, right? It's going to continue to shrink, right? And that's because osmosis is driving the water out of the cell. Okay, and again, you'll probably encounter something like this on the MTEL. All right, last of the organic molecules was the carbohydrates. So monosaccharides, glucose is what we're generally talking about when we talk about blood sugar. Glucose is like a cell's favorite energy source. Cells love it. There's other types of monosaccharides, fructose and galactose, that look similar. These are all monosaccharides. They can be built into polysaccharide chains, right? And examples of polysaccharides are like starch, glycogen, and cellulose. Okay, and then, oh, sorry, nucleic acids is the last one. The nucleotides are the subunits here, right? For DNA, it's A, T, C, and G is the way I remember that. Just so it rolls off my tongue, A, T, C, G. That's adenine, thymine, guanine, and cytosine. RNA is really similar, right? But instead of thymine, right, it's uracil. So the subunits are a little bit different, A, U, C, and G. I'll next present 6 questions, multiple choice questions. I'll start by reading the question, then I'll pause so you can think about the right answer, and then I'll describe the right answer. I'll indicate what the right answer is. All right, so for question 1, a researcher hypothesizes that watching a video on the phases of the cell cycle will be a less effective learning tool than asking students to sequentially arrange cards that describe the phases of the cell cycle, G1, S, G2, and M, using the same level of detail as the video. The researcher separates 48 college students into 2 groups of 24, with an equal number of freshmen and sophomores in each group. One group watches the video, and the second works to arrange the cards sequentially. All test subjects are then asked to write down a description of the cell cycle. Which of the following is the independent variable in this experiment? The answer is C, right? The teaching method. Right? That's what you're looking to see if it has an effect. Right? The completeness of the cell cycle description, right? That would be the dependent variable. How well did the students do? Well, it depends, right? What method did they use? Right, so the independent variable is the teaching method, C. A researcher presents the data from the cell cycle experiment in a bar chart. The student scores versus teaching method. Which will be presented on the x-axis of the graph? In this case, it's C, right, which is what we were just actually kind of thinking about in the last question, right? So on C, or with C, on the x-axis is where you plot the independent variable, right? So that would be the method. I can already envision it, right? You have the video, right, versus arranging cards in a certain way. You have these 2 different methods, right? And then what you're kind of assessing is how well the students describe the process after they've used each method. Right, so that would be the dependent variable. How well do they do? It depends, right? So then that would go on the y-axis. Which of the following properties of water is responsible for the solubility of many molecules in aqueous fluids, such as blood and urine? So these are all interesting and properties of water, right? But only one of them is the best answer in terms of being relevant for why it's the solute of life, the solvent of life, why it's such a good solvent, right? And that's because that it has this polarity characteristic, right? So a single molecule of water has the more negative oxygen end of the molecule, as the more positive hydrogen ends of the molecule. Right, so it has this polarity. It's one molecule with variability in charge across it, so it can interact with other molecules, opposites attract, whether they be positive or negative. Right? So that makes water a great solvent. Question 4, which of the following pose the greatest obstacle to the random passive diffusion of ions across the plasma membrane of a cell? The answer here is A. Right? So if you remember, phospholipids have kind of a charged head, right? Hydrophilic with a phosphate group, but then they have these pairs of fatty acid tails, these hydrocarbon chains, which are not polar, which don't favorably interact with water. They're hydrophobic. So they create kind of this hydrophobic barrier. And ions are very small as singular entities, right? But they're charged and they interact with water. And they're also not energetically favorable to get across that kind of greasy, lipid, hydrophobic internal environment. So ions don't diffuse just across membranes. They for sure need help with proteins that are facilitated diffusers that allow them to cross down concentration gradients, right? Or active transporters like the sodium-potassium pump, right? Proteins do great jobs of moving ions across membranes, but ions can't just diffuse across lipid bilayers. Which of the following treatments will most likely cause cells to burst and release their contents? So this, I can't tell you how many times I've encountered cells and osmosis questions over the last 25, 30 years. Right? This comes up all the time. And for sure, right, this is a case where you have cells that are in a hypotonic solution, hypopop burst, hypo, right? So it's really dilute. So what's water going to do? It's going to follow the salts. In this case, inside the cell, so it'll continue to go into the cell until it pops. Right? Any way you want to think about this to make it work, use that. But this is definitely a circumstance where you're going to get the diffusion of water into the cell, right? Or osmosis is going to drive the movement of water into the cell, which is more concentrated, until an equilibrium is met, right? And that may entail the bursting of the cell. All right, lastly, ACE inhibitors are a class of drugs that lower blood pressure by inhibiting angiotensin converting enzyme. This converts angiotensin 1 into angiotensin 2, a substance that increases blood pressure. To investigate whether drug X is a competitive or non-competitive inhibitor of ACE, a series of reactions were performed in the absence or presence of drug X. And the relative amount of angiotensin 2 produced in each reaction was determined. Each reaction contained the same amount of enzyme, was adjusted to the same volume and buffer conditions, and was carried out for the same amount of time. The results are shown in the figure. Which of the following statements is best supported by the data? All right, so first we'll look at the figure, and then I'll present the options to you along with the figure. Right, but what you're looking at here is a graph. Right? So you're looking at these different reactions along the x-axis. Right? Along the y-axis, you're looking at how much of this angiotensin 1 was converted to angiotensin 2. It's either the absence or presence of drug and an increasing amount of substrate. So take a moment and orient yourself to the data. All right, so your options are, right, drug X is a non-competitive inhibitor because the rate of the reaction in the presence of inhibitor is slower at all substrate concentrations. B is drug X is a non-competitive inhibitor because the enzyme only functions at the highest concentration of substrate in the presence of the inhibitor. C is drug X is a competitive inhibitor. That's because less of the product is formed in the presence of the inhibitor at all substrate concentrations. And lastly, drug X is a competitive inhibitor because the same amount of product is formed in the presence and the absence of the inhibitor at the highest substrate concentration. So the answer here is D. And what I would do is refresh my memory as I'm kind of thinking through this question on what does it look like when an enzyme is doing a job, right, in the presence of competitive versus non-competitive inhibitors. And then hopefully I would remember that a competitive inhibitor inhibits until the substrate level is so high, right, that it can outcompete the competitive inhibitor from that active site. Right? Or to put it another way, you have this enzyme, you've added some drug, and the drug is blocking the enzyme's activity. Until you add so much of that material, so much of that substrate, right, that it displaces that inhibitor, it wrestles into that active site and it starts to get converted. Right? And that's what I'm seeing in the graph, right? So although the drug shown here is 1 millimolar in terms of the difference between those different experimental conditions, although the 1 millimolar is reducing the amount of product, you can see that in reaction 5, right? As you increase the amount of angiotensin 1, the substrate, from 1 to 2 to 3 to 4, by the time you get to 3 or 4 millimolar substrate, right, there's equivalence between the different reactions, right? So then it doesn't really make a difference if there's drug there or not because there's so much substrate, right? So I would say, okay, this is a competitive inhibitor. After the comma, you're getting a description of why, right? And that jives with what I'm seeing and what I know about it as a competitive inhibitor. And then at that point, I would probably go back and look at the other options and just confirm that they're wrong. That something about them is wrong, because if I can't confirm that, then I need to kind of take another look at it. All right, so this is the end of the first workshop. I reviewed the MTEL, the bio-MTEL structure, and then got into the chemistry of biology and molecules a little bit. Again, that was kind of a spot check of some of the discrete kind of topics within those areas. If any of those areas felt very familiar, then that's great. If not, I would probably take a note of them and make those areas to target when you're looking to other resources to kind of bolster your understanding in those areas. In the next workshop, we'll go over subject matter. So it's going to be dedicated to subject matter: cells, organisms, ecology, and evolution. And then there'll be more practice questions. Thanks.